right. Welcome everyone who's joined the event. We're just gonna hang back for a couple minutes here while we wait for some more attendees to join the event. So we will get started in about 2 minutes here. For those of you who have just joined, my name's Nathan, and I am going to be the host for the event today. We will be getting started shortly here, just in 1 minute to wait for a few more attendees, and then we will get started with our webinar on CRISPR-based diagnostics here. Okay, perfect. So welcome to everyone who's joined this webinar. Say it again, my name's Nathan. I am the host for the event. Today, is on CRISPR-based diagnostics. The CRISPR-Cas platform has been developed for rapid, affordable, and ultra-sensitive testing that can be done in-home for point of care or in the field. CRISPR-based diagnostics have broad applications in detecting a variety of molecular targets relevant to human health and will lead to revolutionary breakthroughs. The topics covered today will be an introduction to CRISPR, Cas proteins, and the applications of CRISPR-based diagnostics. Our speaker today is Dr. Clark Shea, who is the Director of Molecular Biology at SignalChem Life Sciences. Dr. Shea has over 2 decades of experience in drug discovery and development. Prior to joining SignalChem, Clark worked at several Canadian pharmaceutical companies where he has made critical contributions to a wide range of therapeutic programs across many disease areas. These include cancer, inflammation, cardiovascular disease, chronic pain, and central nervous system disorders. Dr. Shea has received a PhD in biochemistry and molecular biology at the Shanghai Institute of Biochemistry, Chinese Academy of Science. He later received his postdoc training in the Department of Biochemistry at the University of Washington in Seattle. Clark is an expert in developing various biological assays for target discovery and validation, high throughput screening, and characterization of lead compounds. Clark is currently working at SignalChem Life Sciences, located in Richmond, British Columbia. SignalChem was founded in 2004 and is now a global leader in protein engineering, manufacturing, purification, and assay development. Recently, SignalChem has launched a diagnostics division headed by Dr. Clark Shea. I'd now like to give the floor to Clark to begin his presentation. Thank you.
Thank you, Nathan. Thank you for your kind introduction. And good morning, everyone. Thank you for joining us for today's webinar. My topic today is CRISPR-based diagnostics. I will introduce the CRISPR systems and its applications, the application of CRISPR for molecular diagnostics. I will explain how the assays work and introduce different test platforms. At the end, I will also give you several examples of CRISPR-based assays used for diagnostic tests. CRISPR is the short name of clustered regularly interspaced short palindromic repeats. Two scientists, Emmanuel Charpentier of Max Planck Unit for the Science of Pathogens in Germany, and Dr. Jennifer Doudna of University of California at Berkeley in USA, won last year's Nobel Prize in Chemistry for their discovery of one gene technology, sharpest tools, the CRISPR-Cas9 genetic scissors. Using these, researchers can change the DNA of animals, plants, and microorganisms with extremely high precision. This technology has had a revolutionary impact on the life sciences. It's contributing to new cancer therapies and may make the dream of curing inherited diseases come true. It only took 8 years from the scientists that discovered the CRISPR to they won the Nobel Prize. This amazed lots of people. And from this, you can tell how remarkable this discovery or technology is. CRISPR systems are a fundamental part of a microbial adaptive immune system. Yeah, it is true. Even single-cell microorganism like a bacteria has its own immune system. With CRISPR systems, the bacteria can recognize foreign nucleic acid basis of their sequence and subsequently eliminate them by means of endonuclease activity associated with CRISPR-associated enzymes we call Cas enzymes. When the bacterial viral called the phage invade the bacterial cells, they inject their nucleic acid into the cells. The bacterial processed the viral DNA to generate short nucleic acids with new spacers and integrate them into a CRISPR sequence, the clusterase. From there, CRISPR RNA is formed. When the bacterial cells is infected by same type of phage, CRISPR DNA, the CRISPR RNA can recognize the foreign DNA and guard molecular machinery to target and destroy viral genome. Based on this, apical making biotechnology was invented. It's called CRISPR-mediated gene editing. I believe most of you know CRISPR because of this application. The CRISPR-mediated gene editing requires three components: the guard RNA, called the gRNA or cRNA, CRISPR-associated nucleus, Cas9, and the programmed DNA fragment. Its steps and the procedures are like this. First, Cas9 forms a complex with a guard RNA, then the complex attached to a matching DNA sequence adjacent to a spacer. The complex then cuts the double strands of the DNA. At this point, programmed DNA may be inserted at the cut to achieve gene editing. CRISPR has shown very broader applications, like gene screening at the genome level to investigate function of genes and discover disease-associated genes. Based on the results, human disease, animal, and cell models can be created for target validation and drug discovery. Molecular diagnostics, I will talk this in more details later on. This is a fast evolving area of in vitro diagnostics. Through gene editing, scientists are developing cell and gene therapies. This is now a very hot and exciting area. CRISPR has been used to develop new biosensors. It also has remarkable application in agriculture and animal husbandry. For example, the pathogen detection, crop and livestock genotyping, and their variety improvement. In industrial bioengineering of biosynthesis, scientists use CRISPR to modify the genome of microorganisms, plants, and mammalian cells by synthesizing small or large molecules on purpose. Actually, the list of CRISPR applications is still on expansion. Currently, CRISPR-Cas systems can be divided into 2 classes, 6 types, and several subtypes. The classes of a CRISPR-Cas system are defined by the nature of the ribonucleic protein effector complex. Class 1 systems are characterized by a complex of multiple effector proteins, and Class 2 system encompass a single guard RNA binding protein. CRISPR-based diagnostics utilize the Class 2 systems, which is very simple for creating the assays. The first CRISPR-based diagnostic method that were developed largely used Cas9 variants, which recognize the double-stranded DNA. However, because the Cas9 system, the guard RNA is more complicated and it has a more requirement for the target gene. So it is not simple to develop a detection assay with a Cas9 system. The development of CRISPR-based diagnostics got a significant acceleration since the discovery of novel types of Cas enzymes, referring here the Cas12 and Cas13. The main difference between CRISPR type 2 Cas9 systems and type 5 Cas12 and type 6 Cas13 systems is the ability of the latter 2 systems to trigger non-specific collateral cleavage on the target recognition. Collateral activity involves the cleavage of a non-targeted single-stranded DNA in the case of Cas12 and all single-stranded RNA in case of Cas13 in the solution, which enable the sensing of nucleic acid through the signal amplification and allows various readouts through the addition of functionized reporter nucleic acids, which are cleaved by collateral activity. Here is an example of using fluorescent labeled small probes for detection of nucleic acid target with CRISPR systems. The probe has a fluorophore at one end of the molecule and a quencher at the other end. When probe is intact, there's no fluorescent signal. When Cas guard RNA complex seeks out the target DNA or RNA in the detection assay, the collateral cleavage activity is triggered. The probes are cleaved. Subsequently, the fluorophore and the quencher are separated, and the fluorescent signal is generated. In order to develop CRISPR-based assays, we produced a set of recombinant Cas12 and Cas13 enzymes. You may ask why not only 1 or 2 enzymes, but a set of Cas12 and Cas13 enzymes. I will tell you the reason later on. On this slide, on the left side, it is example of a Cas12 enzyme called AAP Cas12b. Its activity was tested. The yellow bar represents the reaction without guard RNA. There's no signal detected. It is same as for the reactions without target nucleic acid. Once the reactions contain all necessary components, the fluorescent signal was detected. And you can see the signal intensity is clearly related to the amount of AAP12B in the reactions. On the right side, it shows the similar pattern for Cas13 enzymes. In negative control reaction, which is a reaction without RNA target, so there's no signal. Once the target RNA was added, a clear signal was detected. So that's demonstrate all the assays are strictly mediated by recognition of a target nucleic acid by a guard RNA that is complementary to the target nucleic acid. So the assay is very specific. This diagram demonstrated the flowchart of CRISPR-based diagnostics. Cas enzymes and the specific guard RNA together with reporter are added into a specimen containing a target and a non-target nucleic acid. Once the Cas guard RNA recognizes the target nucleic acid, the non-specific nuclease activity is triggered. The probes are cut by a collateral cleavage activity, and a fluorescent signal is emitted and detected by a spectrometer. There are very broader applications of CRISPR-based molecular diagnostics. This technology can even achieve the detection that the current methods cannot carry out. First, it is an excellent tool for diagnosis of infectious diseases by detecting nucleic acid biomarker of virus, mostly RNA-based, and bacterial, mostly DNA-based. It can also detect antibiotic-resistant gene to help doctors to prescribe proper, effective antibiotics to treat infections. For diagnostics of genetic diseases, this technology is sensitive enough to detect even single nucleotide changes. It will also have a profound impact on cancer therapy. Actually, most cancer are treatable if it can be diagnosed at the early stage. So diagnosis or pre-screening of cancer become critical for winning the fights against the cancers. CRISPR-based diagnostics can effectively detect the mutations or modifications of oncogenes. Another obstacle for cancer treatment therapies is drug resistance. CRISPR-based diagnostics can also be used to investigate the drug-resistant gene for providing important information to help doctors to choose effective treatment. There are different CRISPR-based diagnostic platforms. Here are two most advantage platforms. One is a detector platform developed by Nobel Prize winner Dr. Jennifer Doudna's group. They use Cas12 for detecting a DNA target. In the middle, the Sherlock is developed by MIT professor Feng Zhang's group. They use Cas13 instead to detect the RNA target. Recently, the same group also published Sherlock version 2 with increased sensitivity, which can even detect the target and generate lateral flow readouts. It's a more convenient portable assays. You may notice for both platforms, there is a first step called RPA. RPA stands for recombinase polymerase amplification. It is one of isothermal amplification technologies. In contrast to traditional PCR amplification, which requires thermal cycling of 3 or at least 2 temperature, for the denaturing, annealing, and extension to carry out the amplification. Isothermal amplification only need one temperature. Therefore, unlike the traditional PCR, isothermal amplification does not require sophisticated equipment. Like a heat block is enough. For RPA, the technology can be achieved at the 37 degree, so we can even hold the reactions in hand to achieve the amplification. Through this step, the nucleic acid target in the sample is amplified, and as a result, the assay sensitivity is significantly increased. There are other isothermal amplification systems you may heard, such as a loop-mediated amplification called a LAMP. However, because RPA need a lower amplification temperature and has a lower non-specific amplification, right now this technology is used more than others. Here is an example of CRISPR-based test. No doubt, currently the most popular and performed test is SARS-CoV-2 detection. The tests based on both detector and Sherlock platforms are developed. In detector tests, the target nucleic acid SARS-CoV-2 RNA, SARS-CoV-2 virus is RNA virus. The RNA is retrotranscribed to the DNA and amplified. Then Cas12 is used for detection of the target gene. There are fluorescent or lateral flow readouts for the test. Similarly, in Sherlock, there are also two types of readouts: the fluorescent readout and also the lateral flow readouts. Some of diseases are not caused by mutations, gene mutations, but abnormal epigenetic modification. For example, studies have revealed that abnormal methylation of a human genome has a high correlation to colon cancer. And there has been an access for pre-screening colon cancer by monitoring the methylation, human genome methylation. CRISPR-based diagnosis can also be used to detect epigenetic modifications like methylation. In assay, the guard RNA can match normal human nucleic acid target and triggered collateral cleavage of probes that leads to emission of a fluorescent signal. If the gene has methylation and modification, the guard RNA can no longer hybridize with the target. As a result, there will be no fluorescent signal because the no recognition, then Cas enzyme cannot be activated. Comprehensive and simultaneous testing of multiple targets is always desirable in many diagnostic settings, as it can provide more information at high throughput and at a lower cost per target. The design of a multiplexed CRISPR-based diagnostics is to take advantage of the specificity of different Cas enzymes with regards to their collateral cleavage activity towards different nucleic acid sequence, the reporter sequence. This allows multiple nucleic acid targets to be detected by Cas enzymes within one assay. For the example of a multiplex detection showed here on this slide, different Cas enzymes are used for detecting different targets. Their preferred specific probes are labeled with a different fluorophore, such as FAM, text, sci-fi, or fax or hex. Their signal can be detected at different wavelengths. Based on the signal types and intensity, the presence of different targets can be detected in one assay. Now you can understand why at the SignalChem we produced a set of Cas enzymes and are still making more. It is because we want to increase our capacity to develop a multiplexed detection assays. The fast and accurate diagnosis of disease is central for efficient therapeutic intervention and epidemiological surveillance control. I believe we can clearly see this importance from current COVID-19 pandemic. Because DNA and RNA can be amplified from trace amounts, molecular diagnostics based on the detection of nucleic acid are among the most sensitive and specific tests. Currently, the main molecular diagnostic platforms are quantitative PCR, the qPCR, and the next generation sequencing, the NGS. In particular, the qPCR tests play a significant role in the current campaign against COVID-19. However, both platforms require costly equipment and trained personnel. Comparing to those two platforms, CRISPR-based diagnostics has its advantages I summarized in this table. First, it is sensitivity. CRISPR-based assays have excellent anti-interference ability. It is able to amplify and detect a nucleic acid target at a concentration as low as 8 molar, which is 10 to minus 18 molar, from a sample with a very complicated background. The PCR-based assays have a poor anti-interference ability, which can lead to ineffective amplification, often translated into false results. So, like in the SARS-CoV-2 test, you may often hear about the false negative or false positive controls, false positive results. Regarding the specificity, CRISPR-based assay can detect a single nucleotide mutation. To achieve the same level of specificity with PCR-based assays requires a combination of sequencing and other technology. So it is more costly and complicated. Adaptability. Design of primers for amplification in a CRISPR assay is relatively easier. There is no need for lots of optimization, and it is easy to develop and deploy a new CRISPR-based detection assays. For operation difficulty, I have already mentioned that CRISPR-based diagnostics does not require sophisticated laboratory equipment and well-trained personnel. This is particularly important for the test performed in lower income countries and remote areas. Lastly, is the cost. CRISPR-based tests are much cheaper. I'll give you the example here. The current SARS-CoV-2 nucleic acid test costs dozens of dollars and take at least 4 hours to get the result. The equivalent tests with a CRISPR-based assay can only cost less than $1, and it takes as less as 30 minutes to get the result. Constantly, there's a rising need for development of ideal diagnostic platform for a rapid, sensitive, specific, and cost-effective detection of a wide range of pathogens and diseases. It should be user-friendly and able to give instrument-free readouts for early detection of pathogens. I hope through my presentation today, you can agree with me that the CRISPR-based molecular diagnostics meets all the required criteria of ideal diagnosis. So we have every reason to call this the next generation diagnostics. Actually, currently at SignalChem, we are undergoing several projects to develop the CRISPR-based tests. That's all for my presentation today, and thank you for your attention. I think we may still have time for a couple of questions. Thank you.
Hi, Clark, and hi, everyone. Thank you for that presentation. It was very informative. I've been receiving a few questions throughout the event. We do have time for about 2 or 3. Now, for those of you who joined the event a little later on, this will be uploaded, so you will be able to watch the full event again, just not live. Now, we will try to get to all your questions if possible. I would recommend sending in to info@signalchem.com if we aren't able to get to one of your questions that's been posed, or if it hasn't been answered throughout the event. So the first question I have here for you is how to choose Cas12 or Cas13 for developing CRISPR-based detecting assays.
Okay, yeah. The first, it depends on the type of nucleus target, because Cas12 and Cas13 has different specificity. Usually for DNA targets, you just choose Cas12, and for the RNA target, you can choose Cas13 enzymes. But things are not always that simple. Cas12 enzymes used for the CRISPR-based diagnostics targeted the double-strand or single-strand DNA requires a specific protospacer adjacent motif called a PAM site in the target region for double-strand DNA cleavage and collateral cleavage of the single-strand DNA, which is the probe I talked in the presentation. Unfortunately, in some targeted genes, it is very hard to find the appropriate PAM sites, or even sometimes unable to find one. So under such circumstances, the solution is to just use Cas13 to detect the DNA target, because Cas13 does not require PAM site. However, for the DNA target, you need to convert the DNA into the RNA by transcription, and then use Cas13 to detect the targeted gene.
Amazing, thank you. I have another question here for you. Is it possible to develop CRISPR-based detection assay without pre-samplification?
Yeah, yes. My answer is yes. However, it's fully, highly depends on the concentration of nucleic acid target in the sample, in the specimen. When using Cas enzymes without the upstream pre-amplification of the target, most CRISPR-based diagnostics have the limit of detection, LOD, in the picomolar range. This LOD allows for targeted detection when there's like a relatively high concentration of DNA or RNA in the sample. For example, SARS-CoV-2 has apparently high concentration of RNA targets in the early phase of infection. There's about 7 × 10⁵ copies per swab. This allows pre-amplification-free detection. In addition to the highly concentrated microbiome samples like SARS-CoV-2, human genomic DNA and highly expressed human messenger RNA or microRNA have been reported to be detected in assays without pre-amplification. Actually, scientists are still exploring various methods to further increase the sensitivity of pre-amplification-free CRISPR-based diagnostics. For example, by incorporating CRISPR type 3 RNA nucleus into the system, this design led to a fourfold increase in the signal sensitivity. Unfortunately, I don't have time to go to the details. So if you have you are interested in this topic, you can send an email to us, and I'm happy to share the information to you.
Okay, thank you. And once again, that email to send in your questions is going to be info@signalchem.com. We will get to your questions there. We do have time for one more question here before the end of the presentation. One of the limitations of CRISPR-Cas9 system is the potential occurrence of off-target effects. What strategies could be used to minimize off-target effects?
That's a good question. So yeah, it is true. Off-target events cause big concerns for the CRISPR-mediated gene editing and its application for gene therapy. But I don't think it is going to be a big issue for CRISPR-based diagnostics. If you think back on the flowchart of a CRISPR-based diagnostics I just showed you, you may notice there are several specific requirements through at different steps through the process. I'd like to call them checkpoints. Yeah, those can ensure higher specificity of the test. In first step, isothermal amplification, the pair of primers have to be specific to target region. Yeah. Of course, like the traditional PCR, there could be a non-specific amplification occur. However, there is a second checkpoint, the specific matching of a target sequence with a complementary guard RNA for target recognition. Upon the target recognition, the Cas enzymes collateral cleavage activity is triggered. Then the assay comes to the third checkpoint, the specific collateral cleavage activity of different Cas enzymes towards different report nucleic acid sequences. So unlike the process with only one checkpoint, such as a qPCR, with a specific hybridization of PCR primers to target genes, or the CRISPR-mediated gene editing with a specific matching of guard RNA with a target sequence. The multiple step specific requirements leads to a high specificity of CRISPR-based detection assays. Yeah, just like you are signing into like a bank account with not only the password, but also the verification code, and plus the answers to the identification questions keeps a higher security for the account, right? So this is equivalent to keep a higher accuracy or specificity of the assays. Yeah, but this does not mean we don't need to pay attention to wise designing specific application primers, guard RNAs, and reporter nucleic acid sequences. Yeah, we do need to pay attention to those. We also need to test the large amounts of samples to determine the accuracy rate of the assays. If the outcome is not that great, you see a lot of false outcome. then we need to redesign those components. Actually, scientists are making efforts, still making efforts to obtain Cas enzymes variants with a higher collateral cleavage specificity, just by searching in the natural world, or just creating the new one by protein engineering. This can help further increase the assay sensitivity and also increase the choices of Cas enzymes that can be used for develop multiplexed detection assays. Yeah, I hope this answers your question.
Okay, thank you, Clark. Thank you for that Q&A. Thank you to everyone who's attended this event. For those of you who may have missed the beginning of it, again, we will be uploading the entire webinar online. That'll be on our YouTube page. And for those of you whose questions we missed, one last time, that is info@signalchem.com. Once again, I would just like to thank everyone who attended this event. We really appreciate it. And for everyone to have a great day. Thank you.